This lecture is entitled Muscles and Muscle Tissue. Muscle tissue transforms chemical energy to mechanical energy in order to exert a force. With skeletal muscle, this force is applied to the movement of the bones in order to move the limbs, or in cardiac muscle, this force is applied to the movement of fluid throughout the body. In smooth muscle, this force is applied to the movement of sphincters and parts of the digestive system to move food particles from one portion of the digestive system to the other. The prefixes myo, mis, and sarco are prefixes for muscle. So when you see these prefixes, think in terms of something muscular. Skeletal muscles are striated and are voluntary control. That means they're controlled by the conscious mind. And remember, muscles can only pull, they don't push. They can only pull things together, they don't push things apart. You've seen this picture before in histology, but this is a photograph of a skeletal muscle. You see the striations in there. See the function and location of the muscle. This kind of basic histology for the skeletal muscle. There's also cardiac muscle, which is found only in the heart. Cardiac muscle, as you recall, is striated and is involuntary control. In other words, you don't have to consciously think about contracting the heart muscle. It just does it automatically. The third type of muscle is smooth muscle. This muscle is found in hollow organs like the stomach, the urinary bladder, the airway, the respiratory tract. These are non-striated muscles. They're involuntary as well. There are 4 characteristics of muscles that are unique to muscles. Excitability, or responsiveness, which is the ability to receive and respond to a stimuli. Contractility, the ability to shorten forcibly when stimulated. Extensibility is the ability to stretch. And elasticity is the ability to recoil and resume its predetermined resting length after stretching. Muscles have several functions in the body. Obviously, one function is they produce movement. Another function is they maintain posture and maintain body position. They stabilize joints and they generate heat. This is in the form of shivering. When you get cold, you shiver. Those are contractions of muscles to generate heat. Let's look at the way the muscle is organized. First of all, we have this portion here is a muscle cell, also called a muscle fiber. And in the muscle fiber, you see these rods. These are myofibrils. We'll discuss those later, but these myofibrils are contained within the muscle cell or muscle fiber. Muscle cells are grouped together in a bundle called a fascicle. This is a fascicle here. This is a fascicle, a group of muscle cells. And the fascicles are arranged within the muscle as a whole. The whole muscle is composed of several fascicles. So you have whole muscle, fascicles, muscle cell or muscle fiber, and then the myofibrils within the cell. The membrane that surrounds the whole muscle is the epimysium. Remember, epi means above or over. So epimysium is a dense, irregular connective tissue that surrounds the entire muscle. There's a connective tissue that surrounds each individual fascicle. That's perimysium. Again, peri means around. Mys, M-Y-S, means muscle, so that's around the muscle. This is the membrane that goes around the muscle fascicle. The endomysium, you see here, is the endomysium around the muscle fiber, and in between the muscle fibers. This endo means inner or within, and mysium means muscle. This is within the muscle. This is a membrane that surrounds individual muscle cells or muscle fibers. This is made of fine areolar connective tissue, by the way. When we talk in terms of muscles and the attachments of muscles, we refer to an origin and insertion. Each muscle has an origin and an insertion. The origin is the attachment of the bone that's less movable. The insertion is attachment to the more movable bone. like in this case of the biceps muscle attached to the shoulder and across the elbow to the forearm. If the shoulder, most often when you contract your biceps, your shoulder's going to be still and you're bending your elbow. So the more movable joint was going to be the below the elbow, so that would make that the insertion. The less movable joint is going to be the shoulder, which makes that the origin of the muscle. I think more important than knowing the origin and insertion muscle is just knowing where the muscle is attached, and knowing that the muscles pull together, not push. You can derive the movement of the muscle just based on the location of the muscle attachments. Speaking of attachments, there's two types of attachments. There's direct attachments and indirect attachments. A direct attachment is when the epimysium, remember that's the outer layer of membrane around the muscle, the epimysium is fused to the periosteum of the bone. Remember, the periosteum of the bone is the outer membrane of the bone. A perichondrium of cartilage is the membrane surrounding the cartilage. So direct attachment is when the muscle connects directly to the bone. Indirect attachment is when the muscle uses a tendon or aponeurosis to make contact with the bone. Here we see an example. The brachialis muscle is attached to the bone here. This would be an example of a direct attachment. And indirectly, it has a tendon here that attaches down towards the bones of the forearm. So this would be an indirect attachment. The biceps, on the other hand, has a tendon that attaches both at the forearm and also at the shoulder, so it has an indirect attachment at both ends. Both the origin and insertion are both indirectly attached. Now, let's look inside the muscle cell itself. I said these tubules are myofibrils, and that's what gives the muscle the striated appearance. We'll discuss the striations here just in a moment. Here's a close-up of that portion. We're taking this portion here and going to focus in on that, and we have this. So we have our muscle fiber here, surrounded by the endomysium here. It's been cut away before you see inside. These are the myofibrils, which make up the inside of the muscle cell. Notice these myofibrils have bands that are light and dark bands. That's what gives a striation appearance. You see the bands more clearly here, actually. The sarcolemma, remember that's a prefix that denotes muscle. Sarcolemma is just another name for the plasma membrane of the muscle fiber, this membrane that goes around the muscle fiber. Sarcoplasm is a cytoplasm, which is the intracellular fluid within the muscle fiber. Myofibril is a single muscle fiber, has hundreds of thousands of rod-shaped myofibrils running parallel to the length of the muscle fiber. Each of these are myofibrils. They're the components within the muscle cell. Now this myofibril, here's taking one of those myofibrils. Let's look at this myofibril here and blow that up and look at it closer. Here's the myofibril here. And if you notice, it's actually composed of even smaller fibers called myofilaments or filaments. These are filaments here. So the structures from large to small is muscle fiber, muscle cell, is made up of muscle fibrils, which is made up of myofilaments or otherwise called filaments. It's these filaments that give the alternating light and dark bands that form the striations of the muscle. And we're going to look at how these filaments line up and what these filaments are composed of. First of all, there's thick filaments. Thick filaments are made up of several hundred myosin molecules. This is a single myosin molecule. This is several of them bundled together, making a thick filament. So remember, thick filament is made up of myosin. We have thin filaments made of actin. Actin is represented here by the round spherical structures that are in line. That would be the actin. There are two intertwined strands. Tropomyosin is also part of the thin filament. Tropomyosin is represented by this light blue band here, tropomyosin, spirals around the actin and stabilizes the actin. But what it also does, it blocks the myosin binding sites that prevents the thick and the thin filaments from sliding across each other. To give you a preview, the way the muscle works is these thin filaments slide over the thick filaments. They slide relative to each other, and thereby shorten the muscle. The tropomyosin prevents that sliding from occurring. And troponin is another molecule that's a part of the thin filament. That's this yellow triangular shaped structure. It has three regions. One region binds to actin, one binds to the tropomyosin, and another region binds to calcium. So that thin filament is made up of three components: actin, tropomyosin, and troponin. Actin's easy to remember because actin rhymes with thin. Actin and thin kind of sound the same. So you can remember actin and thin filaments. Tropomyosin and troponin are the other two parts of thin filaments. Now there's elastic filaments that are also at play here. The titin is a huge springy protein that acts as an elastic filament. It connects to the Z-disc, and I'll tell you what the Z-disc is in just a minute. That's a part of the structure of the muscle. But the elastic filaments prevent overstretching of the muscle. So when you have when you put these filaments together in the muscle, they have a very uniform pattern. They don't just form randomly, they have a very organized pattern. And let's look at this pattern for a little bit. Here's the thick filaments here in purple. And we have the thin filaments here in kind of orange color, thin filaments. These thin filaments are attached to the Z-disc. That's what I was talking about, the Z-disc. Z-disc would be the end of the structure. This is a sarcomere. Basically, sarcomere is from one Z-disc to the next Z-disc. And that's the functional unit of a skeletal muscle. That's basically, that's the working unit of the muscle. And that Z-disc marks the edge or the boundaries of the sarcomere. Next to it is another sarcomere, so the sarcomere are lined up in series with each other. So you have thick filaments and you have thin filaments that overlap each other. When the muscle contracts, these thin filaments slide along the length of the thick filaments and cause the Z-disc to get closer together, therefore shortening the muscle. But there's certain bands, certain portions of the sarcomere that you need to know. There's the I band, there's the A band, there's the H band, and there's the Z disc and the M line. And I'll tell you how you can remember that. It just shows the striations, just remind you the striations are formed in the skeletal muscle. I have a little animation here that I'm going to play. This is what happens to the filaments when the muscle contracts. Notice how the thin filaments slid and came closer together, and they slid next to the thin filament, next to the thick filaments, but the thin filaments came closer together during that contraction. Let's look at it again. That's in the resting state. Then when the muscle contracts, this is what happens. They come together and it shortens the sarcomere. Okay. Let's look at the components of the sarcomere. First of all, we'll start talking about the Z-disc. The Z-disc is the end point. It's the end of the sarcomere. Just like the letter Z is at the end of the alphabet. And the Z-disc has a zigzag appearance to it. You can't see it because of the bar, but the bars are. See if I could show you another picture. So there's this picture of the Z-disc without the bar in front of it. It has a zigzag. So that's the Z-disc. Z-disc marks the boundaries of the sarcomere. So the sarcomere goes from one Z-disc to the next Z-disc. That's one sarcomere. Now if we look at this space here between the thick filaments, this is called an I-band. Here's what the I-band looks like with the bar removed. It's this space that's between the thick filaments. It's called the I-band. The term I stands for isotropic. The prefix iso means the same, like in the word isolation means lonely, being alone or isolated. Iso means the same. So the I band has only one filament. And that's why it's iso. It's only one filament. It has a thin myofilament. See, there's only thin myofilaments here. There's no thick myofilaments in the I band. I band does not cover over the thick filaments, it just has the thin filaments only. That's I band. Now the middle band, the A band, covers the thick filaments. But it also, has, this is A band here from one end of the thick filament to the other. It also has thick and thin filaments in it. See, the thin filaments are also part of the A band. That's because the A band stands for anisotropic. The prefix "an" in front of a word means the opposite of, like anaerobic is without oxygen, just like aerobic is with oxygen. Anaerobic means the opposite of aerobic. So anisotropic means the opposite of isotropic. Opposite of one is not one, you could think. So the anisotropic band has more than one filament. It has the thick filament and part of the thin filaments in it. So that's why they call it A band. I'm trying to make some sense of how these letterings came about. Hopefully this makes sense. So let's look at the I band and the A band. The H band is the distance between the ends of the thin filaments. The thin filament ends here, and the distance between those two is called the H band. H stands for Healy, which means bright, and it's bright because this is a lighter image, because there's no thin filaments in here between those spots. If I move that bar out of the way, you can see the H band. It just has thick filaments only. So you remove the thin filaments out of the way, and that's the H band. And this slide here shows the M line, which stands for the middle. This is the middle of the sarcomere. This is where the thin filaments, when they contract, they approach towards the M line. They approach from either side. They come towards the M line and shorten the sarcomere. I'll show this animation again, so now that you know the components, you could see this work. So this is resting state, and now the muscle contracts, and the thin filaments come together, and they slide next to the thick filaments. Now the sarcomeres are the functional unit of the muscle fiber. They're the area between the Z-disc. Individual sarcomeres line up end to end along the myofiber like the boxcars of a train. So if each boxcar represents a sarcomere, they lined up in series with each other. When each of these sarcomeres contract, it's going to, if this boxcar was a sarcomere, it would make the boxcar would actually shorten. It would be not as long, it would shorten. And if that shortens a little bit, they all shorten a little bit, you can have a pretty significant shortening of the whole muscle when all the individual components shorten a little. Here's another cartoon showing the same thing. This is our thick filaments, our thin filaments in blue, the Z disc, zigzag, and Z is at the end of the sarcomere, here. Our I band is here. It goes from the edge of the thick filament to the next edge of the thick filament, and it just has the thin filaments inside the I band. Here's an I band on this side. The A band, you can see the A band is the same width as the thick filament. A is anisotropic, so it means it's not a single fiber. It's got the thick filament and has some thin filaments, both thick and thin filaments in the A band. The H zone is this space in the middle. It's between the thin filaments. So this is a relaxed sarcomere. Notice the changes that happen when it contracts. It's not going to show movement, but it's just going to show this contracted state. Here's it's contracted. Notice that the first thing you notice is these thin filaments have come together. They approach towards the M line. They come together. The Z disc, which was way out here before, is now coming closer. So the space between the Z discs gets smaller, and that shortens the sarcomere. And that's why you think this sarcomere is shortened, but all the sarcomere is shortened because muscle contracts as a unit. So all these sarcomeres shorten, and it can result in significant shortening. When these thin filaments come together, that H zone, which stands for bright, goes away. Let me back it up. Here's our H zone. See how this is a bright color? It's a brighter, it's kind of light colored in the middle. When it contracts, like now, the muscle is going to contract. That light zone goes away and it's dark throughout. So those are some changes that occur during the contraction of the muscle. You'll need to know the I band and the A band, the Z disc, the H band, and what happens to these bands when the muscle contracts, because that would demonstrate that you're understanding the workings of the sarcomere. This is what happens when the sarcomere contracts. I bands shorten. The I bands get shorter. The distance between the Z disc gets shorter. The H zone disappears. It goes away. The A band does not change in length. It remains the same length. Then we'll talk about excitability, or the ability to receive and respond to stimuli. Skeletal muscle contracts only under voluntary control. So the first thing to happen to make a skeletal muscle contract is it receives a signal from a motor nerve to start the sequence of events which leads to muscle contraction. This is what excitability is: the ability to receive and respond to stimuli. So there's a nerve that connects to the muscle. The muscle only contracts when the nerve tells it to contract. The nerve uses ions to perform, the muscle uses ions to perform its movements. And what are ions? Ions are charged particles. Examples are sodium, a charged particle of sodium, and negative particle of chloride. Sodium chloride is what salt is. When you put salt in water, it separates into the ion form, and it dissolves in the water, and it's present in ion form. We know the opposite charges attract each other. Positive charges are attracted to negative charges. Negative charges are attracted to positive charges. When opposite charges are separated, there's a potential energy. If you have all the positives on one side and the negatives on another side, and you separate them so they can't cross over, that creates a potential energy because there's a desire for those positive charges and negative charges to come together. This potential energy provides energy needed to create an action potential. An action potential is the energy wave that travels down the length of the muscle fiber that causes the muscle fiber to shorten and contract. Let's look at some of the conditions of the cell itself. Inside the cell, the sodium concentration is low compared to the outside the cell. That's why the outside sodium is written in large letters, the inside is small letters. There's more sodium outside the cell than inside the cell. The opposite is true with potassium. Potassium is more concentrated inside the cell than outside the cell. Hence the larger potassium symbol inside the cell than outside the cell. The inside of the cell has a negative charge compared to the positive charge on the outside of the cell. This is a sodium potassium pump, and I'll go into more detail about that in just a moment. But mainly the slide just shows the initial conditions of sodium concentration and potassium concentration in and outside the cell. Now, ions are they're charged particles and normally can't go through the cell membrane. The cell membrane acts as a barrier to prevent these ions from coming from inside the cell to outside the cell, or vice versa. There are proteins that span the lipid bilayer. So there's proteins that cross this membrane layer. The proteins act as channels, and these channels can allow the ions to cross from inside to outside the cell. If it weren't for the channels, the ions wouldn't be able to cross, but these channels allow crossing of ions from inside to outside the cell. There's specific channels for sodium, there's specific channels for potassium and other ions. Let's look at some of these channels. There's leakage channels that are always open. They don't have the ability to close, they're always open. And there's channels that are gated, that open and close, just like a gate to a fence can open and close. You open the gate to let the cows out. You close the gate to keep the cows in. A gate opens and closes. There's two types of gated channels. There's ligand gated channels and voltage gated channels. A ligand gated means the channel opens when a specific chemical or neurotransmitter binds to the gate. Here's a picture of a ligand. Here's our gate here with a place waiting for a receptor for chemical to bind. When that chemical binds, it causes the gate to open up. Once it opens up, then ions can flow through that gate to go from outside to inside the cell. This is a ligand gated channel. Now there's voltage channels also. The voltage channels open and close based on the charge of the inside and outside of the membrane. I told you before that the inside of the membrane is negatively charged, the outside of the membrane is positively charged. If that condition were to reverse, such that it is positive charge on the inside and negative charge on the outside, that's called depolarization. Polarization is when the negative charge inside and positive on the outside. You have negatives and positive charges are separated from each other. When that reverses, it's called depolarization. It's negative on the outside and positive on the inside. The depolarization causes the channel to open, allowing the ions to flow through the channel. In this case, the sodium goes from outside to inside the cell. So it's important to know the difference between ligand-gated channels and voltage-gated channels. Another word for ligand-gated is chemically gated channels. This just shows the same type of thing. How the channel is closed when the ligand binds to the channel, opens it up, and allows the ions to flow through. Here's a voltage-gated channel. Have the voltage is polarized here. Have it's negative on the inside, positive on the outside. When that changes, such as negative on the outside and positive on the inside, it opens up the channel, allowing the ions to flow through. Now, when these gated channels are open, this is the same for a ligand-gated or a voltage-gated channel. When the gated channel is open, this allows the ions to flow freely from one side to the next side. Well, remember, outside the cell, there's a lot of sodium ions. Inside the cell, there's not many sodium ions. So one thing, the sodium in a concentrated area is going to be attracted to the less concentrated area. So that's called moving down the chemical gradient. So the sodium goes from a high concentrated area outside the cell, travels through the channel to the inside of the cell where it's less concentrated. There's also electrical force. Remember, the sodium ions are positively charged, but the inside of the cell is negatively charged. So this positive ion is attracted to the negative environment inside the cell. So both the electrical differences and the concentration differences drive these positive charged particles inside the cell. In the case of the muscle, it's going to be sodium ions. Let's look at the resting membrane of the muscle cell. It's about 90 millivolts inside the cell compared to outside the cell, and the membrane is polarized. As I said, polarized means it's negative on the inside and positive on the outside. Here it illustrates polarized membrane, negative on the inside, positive on the outside. Negative on the inside of the cell membrane, positive on the outside of the cell membrane. If you look at this portion of the membrane, it blowed up to this structure here. Here we have a leak channel where it allows, these channels are always open. It allows potassium to diffuse out. There's more potassium leak channels than there are sodium leak channels. There's more potassium leaks out than sodium leaks in. So there's more, and both sodium and potassium are positively charged. So if more positives are going out, that's why the inside of the cell is negatively charged, because it's losing the positive charges to the outside. And there's a sodium-potassium pump. It pumps out 3 sodiums in exchange for bringing in 2 potassiums. So the net effect is you have 3 charges out, 2 charges in. That creates a negative charge on the inside, and it helps to maintain the negative charge on the inside. It also helps replace the potassium that's leaked out. It pumps potassium back in to restore the balance of potassium inside the cell and outside the cell. And that's the condition at rest. This basically says the same thing. The resting membrane potential is a result of different ion concentrations inside and outside the cell. The fact that more potassium leaks outside the cell than sodium leaks inside the cell. And the sodium potassium pump transfers 2 potassiums into the cell in exchange for pumping 3 sodiums outside the cell. So let's look at the nerve that causes the muscle to contract. This is the end of a nerve axon, a motor axon. So we'll discuss nerves in the next unit, but just say that this nerve is connected upstream. Maybe the head, the body of this nerve may be up in the brain or some other place, and it sends this axon down. And this is the terminal portion of the axon, the part of the axon that interacts with the nerve. This is a little bit of what we have here. There's these vesicles filled with chemicals that are called neurotransmitters. These chemicals were made up in the cell body, way up in the brain or wherever the cell body is located, and they've been transported down to the end of the axon, waiting to be released. So they just kind of accumulate here in the axon, distal end of the axon. An action potential comes down the nerve, and we'll talk about action potentials later, but just for now, just take it on my word that action potential comes down the nerve and it activates activates calcium channels, which allows calcium to flow into the cells, just like we saw with the sodium going into cells. The calcium rushes into cells. So the action potential arrives at the axon terminal of the motor neuron. Voltage-gated calcium channels open. Calcium enters the axon terminal down its electrochemical gradient. Now the calcium has an effect on these vesicles. Calcium causes these vesicles to fuse with the cell membrane of the junction between the nerve and the muscle, called the neuromuscular junction. Once they fuse, it opens up so that these neurochemicals can leak out into the space called the neuromuscular junction. Acetylcholine is the neurotransmitter that's involved with this procedure. Acetylcholine then leaks out of these vesicles into the neuromuscular junction. They cross the synaptic cleft. That's basically the space between the nerve and the muscle, and it binds on a receptor on the sarcolemma of the muscle. Remember, sarcolemma is a muscle membrane. Sarco means muscle, lemma is the membrane, sarcomembrane. There's these receptors. These are ligand-gated receptors. And the acetylcholine binds on these ligand-gated receptors. It causes those receptors to make the channel open up, like we see here. Here it shows a close-up of the channel. So the acetylcholine is bound to the channel, opens up the channel, and allows all the sodium to rush in and the potassium to rush out. Now, before the channel opens, remember it's negative on the inside, it's positive on the outside. Negative on the inside, positive on the outside. But the effect of the acetylcholine does not last forever. The acetylcholine is degraded by an enzyme called acetylcholinesterase, which breaks acetylcholine away and disables the acetylcholine, so then the channel closes again and it stops that flood of sodium ions from coming in the cell and potassium ions from leaving the cell. Here's a little video that may illustrate this better.
Nerve impulses, also known as action potentials, travel from the brain or spinal cord to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron to a skeletal muscle fiber. The site where a motor neuron excites a skeletal muscle fiber is called the neuromuscular junction. This junction is a chemical synapse consisting of the points of contact between the axon terminals of a motor neuron and the motor end plate of a skeletal muscle fiber. The events at the neuromuscular junction occur in 7 coordinated steps. Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal. Step 2. Voltage-gated calcium channels open and calcium ions diffuse into the terminal. Step 3. Calcium entry causes synaptic vesicles to release acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels. Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative. Step 7. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma. Neural transmission to a muscle fiber ceases when acetylcholine is removed from the synaptic cleft. This removal occurs in two ways. 1. Acetylcholine diffuses away from the synapse. 2. Acetylcholine is broken down by the enzyme acetylcholinesterase to acetic acid and choline. Choline is then transported into the axon terminal for the resynthesis of acetylcholine.
We hope that explanation would help clarify some of the steps at the neuromuscular junction. Now let's look at contractility, the ability of the muscle to shorten when stimulated. Here we go back to our neuromuscular junction. And at this point, the vesicles have released the acetylcholine. Acetylcholine has opened up these channels so they have the sodium ions rushing in, the potassium ions coming out. More sodium comes in than potassium goes out, so it makes the inside less negative. And that's the conditions that we see here. At the resting state, the membrane is polarized. That way, the negative charges on the inside, the positive charges are on the outside of the cell membrane. But once those channels have opened up, then that polarity reverses and depolarization occurs. Depolarization results in the positive charges on the inside and negative charges on the outside. And that opens up these voltage-gated sodium channels. Voltage-gated sodium channels open up. Sodium now comes in this channel. This is a different channel than this. These are ligand-gated channels. These are voltage-gated channels. Two different types of channels. But both of them allow sodium to rush into the cell when open. It has sort of a domino effect. One channel open up, letting sodium come in, depolarizes membrane next to that, which is picked up by the next voltage-gated channel, which allows it to open up, and then it depolarizes the next voltage-gated channel. So there's just a sequence of one channel after another opening up and depolarizing, just like dominoes falling one at a time. Eventually, the sodium channels inactivate and close, and the voltage-gated potassium channels open up, and that allows, first of all, it stops sodium coming in and allows potassium to exit, which gets rid of some positive charges, which makes the membrane go back to negative on the inside. So it basically polarizes or repolarizes the membrane. That's why it's trying to say stage is called repolarization. Okay, that's what's going on at the neuromuscular junction and at the muscle sarcolemma of the muscle. Here's our sarcomere again. Here's our Z line or Z disc, A band, I band. There's a sleeve that's inside the muscle cell that surrounds these myofibrils called the sarcoplasmic reticulum. And look at the sarcoplasmic reticulum. It's just a storage location for calcium ions. Calcium ions are stored in here, and upon a certain signal, they're released. The sarcoplasmic reticulum has an end called a terminal cistern. It's right here. It's next to a T-tubule. A T-tubule is basically a part of the sarcolemma that dives down into the cell. It's a tube that goes down to the inner portion of the muscle. It's made of the same material as the cell membrane of the muscle, of the sarcolemma of the muscle, but just an invasion of that sarcolemma down into the inner workings of the muscle. A triad is a T-tubule flanked by two terminal cisternae, form a triad, the three components. This shows a close-up of the same thing. You can see the triad better, the T-tubule, the terminal cisternae, the sarcoplasmic reticulum, all this blue material here, sarcoplasmic reticulum. So here we have our nerve and neuromuscular junction, and we have an action potential of the muscle that started here. The sodium comes in these channels, they depolarize the membrane next to it, open up other voltage-gated sodium channels, and they propagate down the sarcolemma till they reach a T-tubule, and then that wave of depolarization not only goes on and goes down the muscle, continues down the muscle, it also goes down in the T-tubule and goes into the inner side of the muscle. It goes down deep inside the muscle. So that's why the T-tubules, they're connected to the sarcolemma on the outside, and they bring the action potential down into the cell. Here shows the action potential going down into the cell. Now these T-tubules have calcium channel, voltage-gated calcium channels, so that when this membrane is depolarized, it opens up calcium channels. These calcium channels allow the calcium from the sarcoplasmic reticulum to leave the outer sarcoplasmic reticulum and go into the inner workings of the cell. So now we have a condition where we have calcium dumped into the inside of the muscle cell. Calcium ions are released. Now, remember we talked about the thick filaments and thin filaments. We have our thin filaments here, our thick filaments here. When we talked about the troponin and tropomyosin, troponin had three binding sites. It had a binding site to the actin, it had a binding site to tropomyosin, it had a binding site to calcium. If you look back at that slide, I mentioned that it has a binding site to calcium. Here's where the calcium comes into play. Calcium then binds to the troponin. See if I have this is a copy of the slide we saw before. The troponin has one region binding to actin, one region binding to tropomyosin, a third region that binds to calcium. So the calcium binds to the troponin. Troponin changes the tropomyosin, so that now the tropomyosin no longer blocks the actin from interacting with the myosin. So the myosin and actin can get together and slide across each other. And that's what happens, is they slide relative to each other. It's like the thin filaments slide over the thick filaments. So it goes from here, it slides over. And that sliding is made possible by the fact that the calcium was released that triggered this sliding movement. This diagram just kind of summarizes all that we've talked about. Phase 1 is the motor nerve stimulates the muscle fiber. An action potential comes down the nerve axon and arriving at the neuromuscular junction. Acetylcholine is released. It binds on receptors to the sarcolemma. The ion permeability of the sarcolemma changes. Local change in the membrane voltage or depolarization occurs. Local depolarization ignites the action potential in the sarcolemma, and it follows a chain reaction. That action potential travels across the entire sarcolemma and down the T-tubules. That causes the sarcoplasmic reticulum to release calcium. Calcium binds to troponin. The myosin binding sites on actin are exposed, and the myosin heads bind to actin, and the contraction begins. So that's basically the steps from the motor nerve contraction, stimulating the nerve muscle, to the actual muscle contraction. Hopefully this little animation may help explain it a little bit better.
Typically, a single motor neuron arising in the brain or spinal cord conducts action potentials that travel to hundreds of skeletal muscle fibers within a muscle. The sequence of events that converts action potentials in a muscle fiber to a contraction is known as excitation-contraction coupling. If we look at a single muscle fiber, we see that an action potential travels across the entire sarcolemma and is rapidly conducted into the interior of the muscle fiber by structures called transverse tubules. Transverse, or T-tubules, are regularly spaced enfoldings of the sarcolemma that branch extensively throughout the muscle fiber. At numerous junctions, the T-tubules make contact with a calcium-storing membranous network known as a sarcoplasmic reticulum, or SR. Where it abuts the T-tubule, the SR forms sac-like bulges called terminal cisternae. One portion of a T-tubule plus two adjacent terminal cisternae is known as a triad. The membranes of the T-tubule and terminal cisternae are linked by a series of proteins that control calcium release. As an action potential travels down the T-tubule, it causes a voltage-sensitive protein to change shape. This shape change opens a calcium release channel in the SR, allowing calcium ions to flood the sarcoplasm. This rapid influx of calcium triggers a contraction of the skeletal muscle fiber. Thus, calcium ions are responsible for the coupling of excitation to the contraction of skeletal muscle fibers.
Okay. Let's look at some different terms of what a motor unit is. Each muscle fiber has at least one motor nerve that contracts to cause that muscle fiber to contract. But each motor nerve can stimulate more than one muscle fiber. So one motor nerve can stimulate multiple muscle fibers. A motor unit is the motor neuron and all the muscle fibers it supplies. So there's a small motor unit. If it just has one muscle fiber it contracts. There's some that contract multiple muscle fibers. That's a large motor unit. But a motor unit is the motor neuron plus all the muscle cells that it causes to contract. Here's another diagram of a different motor unit. It shows the motor nerve coming out here, innervating these different muscle fibers. This motor unit 1 innervates and causes certain muscle fibers to contract. Motor unit 2 causes other muscle fibers to contract. So this shows two motor units. Now let's look at a single twitch. A twitch is the simplest muscle contraction. It's basically the contraction of a single muscle fiber. So the stimulus is given to the muscle. It takes a few milliseconds before all the channels open up, and then once the voltage gated channels open up, and the sarcolemma is depolarized, and the way the depolarization moves down into the T-tubules, it moves fairly quickly, and it generates this curve. This curve is tension, or tension pull on the muscle. The higher the curve, the more the tighter that muscle is pulling. This is time on the x-axis. The y-axis is the tension in the muscle. So it takes a few milliseconds to build up tension, but then the tension mounts pretty quickly, and then during the period of relaxation, the tension gradually returns back down to normal. There's three phases to this. There's the latent period, the period of contraction, the period of relaxation. Let me back up and show you. The latent period is this first initial period. The period of contraction, the muscle contracts during this period here, and the period of relaxation is the longer period. And this shows what goes on in each of these periods. You can read that and understand that. Note the muscle contracts faster than it relaxes. Now there's graded muscle responses. The muscle twitches that we just talked about are brief, jerky contractions of a single muscle cell or fiber. But that's not the way our muscles normally contract. Normal, healthy muscles contract smoothly and vary in strength. To get from this jerky muscle twitch to a smooth muscle contraction, there's conditions called graded muscle responses. These are achieved by either changing the frequency of stimulation of the muscle fibers or changing the strength of the stimulation. Let me show you what I mean. Here's our single twitch. This is one stimulus, one twitch. We have tension built up to a certain point, and then it relaxes. Let's say before it completely goes back to zero, we stimulated that nerve again. Maybe we shock the nerve with electrical current and stimulate that nerve again. What happens is it starts the first stimulation, it causes contraction, and then it starts to relax. Before it relaxes zero, we stimulate it again, and it causes more contraction, and it reaches a higher tension. As it starts to fade down, we stimulate it again. Boom. We have more tension, more contraction. We let it fade out and stimulate again. Boom. More contraction and more tension is developed on the muscle. So we're gradually building up more and more tension by each consecutive stimulation of the nerve, of the motor nerve. Now, if we really want to be mean, we could just apply the stimulation so fast that it never does get into, it never can relax. The muscle is always contracting. This would be very painful for the person, but that would be what that happens. That's called tetanus, or complete tetanus, when the muscle is contracted fully for the whole length of the time that the muscle is being stimulated. Now, that was changing the timing of stimulation. We can also change the strength of stimulation, and that involves what's called recruitment. When you first stimulate a nerve, it activates the motor units that are more likely to, that are easier to activate. The more, if more muscle tension is required, then other motor units are recruited. Other motor units are recruited. This shows the basic function of recruitment. Motor units with the smallest muscle fibers are recruited first because they are controlled by the smallest and most highly excitable motor neurons. Motor units with larger motor fibers are recruited as stimulus intensity increases. The largest motor units are activated only for more powerful contractions. So when you want to have more force from a muscle contraction, you start with the smaller motor units and then gradually build up to large motor units based on which motor units are recruited. This shows the same thing. The motor units, one recruited, the small fibers, the medium fibers then recruited, and large fibers recruited. And the combination of this adds to the tension to the muscle by the recruitment of different motor units. Let's look at something a little bit maybe more basic, just the types of contraction. Isotonic contraction is a muscle changes in length and moves a load. Isotonic contractions can be either concentric or eccentric. Concentric contraction involves more muscle shortening when contracting. For example, picking up something with the biceps, picking up this weight. The muscle shortens when the muscle contracts. Eccentric contractions would be a contraction, but the muscle's lengthening. Let's say you've done your curls and you've exercised your biceps and you're fatigued and you try to hold the biceps in a flex position, but the weight that you're carrying just is overwhelming the muscle, and it gradually comes back down. Even though you're contracting, you're pulling against it, you're contracting the muscle, but yet the muscle's lengthening. That's what eccentric contraction is. An isometric contraction is when your muscle contracts, but there's no change in length. The muscle doesn't, the joint doesn't move. It's just the muscles contract. This is an example. If you were trying to lift up a car with one arm, chances are you're not going to move the car, even though you're contracting the muscles. Your muscles are contracting, but there's no movement. That's a bit isometric muscle contraction. Muscle fatigue is when the muscle loses its ability to contract despite continuous stimulation. This can occur with ionic imbalances if the levels of potassium and calcium are down. If in prolonged exercise you can use up these electrolytes. But muscle fatigue is when the muscle cannot contract despite the fact that the muscle is stimulated. This is probably the most difficult of the lectures the students have time with, because there's a lot of complicated material and a lot of technical information given on this lecture. So this is a lecture that's always good to review and to probably look at several times to try to have it make more sense. The first time you see this, it's going to be over most people's head. It's going to be confusing, but each time you listen to it, it should make a little more sense each time. So I'd encourage you to review this again, and if you have questions, certainly we can answer them in class.